So I think I'm back again. Hi, hello everyone. Hi, hello, and welcome again to another Saturday live stream after a short break. I'm back again. Let me simply get some sorted things sorted. Out again over here. Everything hopefully seems to be working. Chat works. Yeah, and after a short break, after 2 weeks of not doing a live stream, I just want to say hello again. And today I've prepared again a couple of little mini projects that I would like to share with you. I see that some of you have already started to join here. So hello from Florida, the United States. Jesse, ever smaller, from England. Hi again. I'll be showing some of the pictures that you've sent me. Thank you. Okay. Hello from Athens, Greece. From Poland, from Germany, from England, from the Philippines, from Poland, from northern France. Really nice. Okay, so just a very brief, the first couple of minutes, just a couple of editorial, some general words here, and then I want to tell you a little bit of some of the things that I'm doing today in the next 1.5 hours. Okay, so first of all, very briefly, I'm back, as you've seen. So I spent a couple of days, a full week actually, on the Canary Islands, which is on Tenerife. It's in the Atlantic Ocean. Spent there a week with my family, and I basically did a little bit of microscopy there because I took along a USB microscope, which I have over here. And I already made a video, by the way, so maybe some of you have already seen a video, which I just uploaded a few days ago. And I put a little sand sample here, also under the microscope, which I would like to show you. I made a couple of videos here, just as a little disclaimer at the beginning, because it's really important. It's actually taking along long sand samples. I did not take this sample along deliberately. I have to be very clear. It's a very small amount, and I actually found that in the bathing suit, in the swimming suits, in the seam of the swimming suit, there were actually some sand grains, which were these here, and that's what I took along. You have to be really careful with taking along sand samples and rock material and so on, because that can actually, it's not allowed for environmental protection, and I support that. But luckily, afterwards, I did not take anything along deliberately, but afterwards I actually did find in my shoe and in the seam of the swimming suit a couple of small amount of sand. And of course I wanted to save that immediately, and I want to show that to you, because what you see here in the center actually is quite nice. Maybe some of you recognize this. This is a spine of a sea urchin. So we're going to have a look at this a little bit. Okay. Okay. There are a couple of suggestions here. Every now and then I will. Every now and then I'm going to read some of the comments. If you have any specific questions, or if you have any specific comments that you want to have, please feel free to post them in the comment section, and at the end I would like to respond to specifically the off-topic questions here. Okay, so very briefly, also another organizational thing here that I would like to talk about today, simply to give you a little bit of an overview. Here, these are bacteria. I would like to do a little bit of staining as well. This is a little bit also if you've seen my video, which I also published some time ago, on the bacteria found in a kitchen sponge. These are actually kitchen sponge bacteria, and I would like to stain them. So I just did a trial here, and it works quite well. So I want to show this to you as well. And I also have a water sample which I just collected today. This is again from a bird bath. I just wanted to have another look at this. So there are Hematococcus algae in here. Again, I already put this sample, not this specific one, but one of those bird bath samples, also under the microscope a couple of weeks ago, and I want to try again, which is a difference right now. And last but not least, something totally different that I would like to do, something that I have not shown you yet, but I brought along a little publication here. It's it looks like this. I would like to show you. This was actually a very short decision. This here is my master's thesis in bacteriology. which I've written this over 30 years ago, and I just kind of rediscovered it again in my bookshelf, and I just want to show to you what I've done at that time, how I analyzed some bacteria. Might be also a little bit, yeah, it's not directly a little bit. I mean, I've got some bad photographs of bacteria that I found here, but I also would like to briefly show that to you because some people might also be, interested in that. Okay, so I'm going to start off. I'm going to quickly read again over here. Hello from Kolkata. This is a suggestion. Basically, not that 15-minute segments maybe are a little bit too short. This is also something that I sometimes feel. I felt a little bit that sometimes I spent a full 1 hour on one topic, which I found a little bit long sometimes. It kind of ended up dragging it out, so I decided to split it up into 15 minutes. Yeah, it depends, I think, a little bit on the topic. Yeah, hello from NC, North Carolina. Okay. I just read here. I tried to find such spines to buy online, but never managed to find any. Ah, okay. Yep. Yeah. Greetings from the UK. Okay. And to mass cultivate. Hematococcus. Okay. Yeah. So what I'm going to do now is again, Ever Smaller. You might want to check out the YouTube channel. Ever Small Jesse actually sent me a couple of pictures that I would like to share with you, and maybe Ever Smaller, maybe you can comment on that a little bit. These are some, basically larvae of some shrimp, if I am correctly informed. So this is a little bit of a video here. Looks like dark field. Looks very nice. Thank you very much for sending me those pictures. This seems to be the egg of one of those. Simply connected them together here. And this one over here is actually a I allowed myself to stitch it together. There were 3 different pictures. This seems to be oblique illumination or DIC. Okay, also very nice. So these are basically larvae of crustaceans. So sometimes you also find so-called Nauplius larvae in a water sample as well. And I kind of like this a little bit, because they look nice in dark field. Okay. There's also a comment here. Did you know that it's illegal to collect sand samples from Italian beaches? Yes, I knew that. And tourists have already been fined very high amounts. Yeah, but not only that, it's important for environmental protection. So I knew about that. And as a matter of fact, it is also illegal to collect rocks, and especially even from areas that are not protected. So I would like to simply say this. I support this generally, but this sand sample here was basically something that I took along accidentally. Okay, it's actually one of the reasons why I also took along a USB portable microscope to do on-site microscopy there. Yeah, so Ever Smaller. So these are pictures that are running around right now in the loop. So it's 400 nanometers across, which is 0.5 millimeter almost. Okay, fairy shrimp. That's how they're called. They're like very large freshwater sea monkeys or brine shrimp. They get huge though. Yes, I've also done this. I might try it again. Maybe you've heard you can buy online, especially for children, so-called sea monkey eggs. These are tiny brine shrimp, and they're also sold in aquarium shops as for food. Because some people who keep an aquarium, they breed them for food. And yes, it's oblique illumination. I wish DSC. Yeah. So this is to my BH2, Olympus BH2, very nice microscope. Yeah. So this is something that looks very nice, and it also shows that if you have basically dark field, so if you have a dark field patch stop, then you're able to see them quite well. Yeah, I highly recommend getting fairy shrimp instead. They are so much cooler, in my opinion. Oh, that's interesting. I don't know. I guess you can maybe also buy fairy shrimp eggs. I suppose maybe that they're also being bred as food for aquariums. I don't know. But if they are basically larger, then of course it might be also easier to observe them. Yeah, but I did try a little bit and experiment around with those tiny sea monkeys, but they are fairly tiny. Yeah. Yeah, so it looks very nice. So something that, yeah, and this one again, I allowed myself to stitch it together. Yeah, the video of the sea monkey toys on YouTube is pretty interesting. Yes. So let me go back. So thank you very much. So if you have also some pictures to send to me that you allow me to share, then that would be, the community would be grateful. Yeah. So I read again. The ones you are looking at are fairy shrimp, not brine shrimp. But yes, they are very awesome. My video on my channel shows them when they are fully grown. Okay. Yeah. So please do visit the YouTube channel of Ever Smaller. Okay. There. And if you yourself, anyone else also has YouTube channels that you would like to, I don't know, maybe put the link into the chat, please feel free to do that. Right. As long as it's microscopy related, obviously, or biology related. Yeah. So absolutely, I have absolutely no problem, and I'm quite sure that yeah, the community is going to be happy about that. Okay, so let's go back to the desk view over here, and what I would like to do now is yeah, I'm going to now show you a couple of videos, and I'm going to comment them. So a couple of holiday videos with a little bit of microscopy. Okay, so I prepared this video over here. So this is some of the clips you might have already seen in one of my my other videos that I published. Okay, so this is basically, yeah, I'm going to pause every now and then. I'm not looking at sand right now, but at some interesting white stuff that I found on a banana plant. Okay, so what I've done here is very briefly, I need a, don't I have an arrow over here? That's a little bit, where's the arrow? Oh, here it is, just a second, yeah. Yeah, so use this obviously USB microscope and directly connected over the cable to my mobile phone. So I was recording directly into the mobile phone. Okay, so highly portable. So that's what's kind of, that's of course the idea here. And I want to show you a little bit also one of the limitations of those this microscope. So this is what I found on the bottom side of the banana leaf, and it looks like this here. And I did some research, and I thought, isn't it some kind of a fungus maybe? But it's not. It's some so-called, I think they call it white flies. I don't know what this is, but these seem to be some kind of insects here. Okay, yeah, there's a comment here. I was just watching the video when you bought this microscope 5 years ago. It's a different one. It's a different one. Okay, just for clarification, because I made some. It looks very similar, but this one actually is has a higher resolution and was slightly more expensive. It looks the same, but it actually has a slightly better quality. Okay, just because there has been a comment right now. Okay, so this is now using the microscope. You see that there are these dots here. I think this might be, and also in the wood there was this white fluffy thing here, and I don't know, almost looked a little bit like a cocoon. But one of the limitations that you already see here with this microscope is, yeah, okay, the delay maybe that you have is that the dynamic range, unfortunately, is not so high. What does this mean? This means actually that you look at the white area over here, you don't see a lot of detail. It's not only because it's out of focus, but you do not see a big difference between the bright and the dark areas. That is generally a problem with those microscopes, that if the color is relatively uniform, you see everything kind of blends in. Yeah. So that's the thing a little bit. And you see these seem to be some kind of fibers here from this insect. I've actually, I'm not showing this to you here, but I've actually later on also found one, and I could actually see, look how messy it is. Yeah, little tiny things that actually appear to be a little bit like eggs or something like that. Yeah, so this is just something that I kind of wanted to share with you, because that's something I did not really know. Yeah, again, the video is looping right now. You see me here with a USB microscope for those of you just joined in, yeah, directly recording. So it was a little bit, how do you say, an experiment on how well you can do a little bit on field microscopy this way, yeah. And again here, on the bottom side of the banana plant, this white fluffy thing here, unfortunately, oh, thank you very much. There's been a donation here. Thank you very much. Okay, so yeah, and again here you see that you don't see a lot of color distinction over here. It looks like a white blob, and that's again the problem of the limited dynamic range, and maybe it's a little bit also overexposed. So I'm going to show you now a second video. And this is actually one of the nicest beaches here, which we visited. Actually, really one of a very beautiful beach, but the sand over there looks white. And this is actually, and also, now this was on site. Yeah, simply a couple of, took 3 pictures, and that's basically how it looks like. Yeah. And I just also want to show you something that I've seen quite a bit here. Let me pause this, and again with an arrow, because that's something that I've also seen. Those fibers here, don't worry, I think not microplastics. I spread some of the sand grains on my, how do you say, on the towel, on the blanket that I was sitting on. And that's basically the reason why you see those fibers here, so not microplastics. But what I've seen also quite recently a lot is what appears to be like glass fragments. Not everything that's so transparent is necessarily glass, but yeah, I've seen that there's more and more also sometimes glass fragments inside the sand. Yeah, so just wanted to also, and the interesting thing about this beach is, and where do I turn this on again? The interesting thing about this beach is that this is actually, the sand is not from the place over there, but this was actually, I did some research in 1973, so over 50 years ago. They did some beach, they made the beach there, so they transported in large amounts of Sahara sand from the Sahara Desert and dumped it there, and to make a nice white beach. Because on the Canary Islands, what you've got mostly is volcanic sand, which is very dark. And you can even see in here that some of the sand grains in there indeed are black. So you see that some of over time there was some black sand grains got mixed into the sand. Okay, so it's simply also a little bit of how do you say, proof of concept. If it's possible to do microscopy on site. The biggest problems that I had with this is that it's kind of unstable, because if you're holding the microscope in soft sand, it's kind of very shaky. Right, that's why I took pictures. So you actually have to use some kind of either you press the USB microscope against the blanket. Where is it? Yeah. Here it is. You kind of for stability. Yeah, but if you kind of hold it like this, it's very shaky. Yeah. So I'm going to read again some of the things here, because I've seen that some comments. Okay. Yeah. Okay. Yeah. Explore the microcosmos. Yes. There are some links in here. Please visit the channels of all of the other people. Yeah. So calcium deposits from the water could be. I'm just reading. If there are any questions, thank you again for the donation. Okay. I've looked green, yellow. I'm just reading. I'm just skimming a lot. Yeah, a little bit on whether there is some comment for me. So there is a third video that I want to show you, and this is now the actual on a natural beach. Okay, this is actually where which the sample that I'm also going to show you right now, and this is now unedited. An unedited picture with a USB microscope, and actually I thought that the contrast was quite nice. So I actually feel that the quality is fairly okay, but you see it's a little bit shaky. Because I was kind of trying to hold the microscope and stabilizing it. The depth of field might be a little bit of an issue. All right. But generally, it kind of works. So my suggestion is if you don't have a stereo microscope, and if you don't, it does not give you a stereoscopic view, obviously. So here, that's again the blue thing in the background is again the blanket, the towel, right? That was on. If you if you don't want to spend any, and if you just want to get a little bit, you're kind of want to experiment a little bit with epi-illumination and something that goes a little bit into the direction where it looks like stereo microscopy, you actually might want to try out one of those. Yeah. Again, this is not a replacement for a full stereo microscope, obviously, but at least it gives you an image that's similar. Okay, so Explore the Microcosmos is asking, I had done some microscopic samples I found in amber. Is it something you might consider trying? Oh, I mean, of course, this is, I'd have to get some of the amber. I would definitely not cut it open, right? But if the amber is sufficiently transparent, right? Maybe it's possible to actually focus through the amber to look at whatever enclosures there are in there. Huh? What was the white thing that you looked at again? The one at the banana tree. Where is this again? That's this one over here. I think that these are this white stuff here. I did some research and they call it white flies. These seem to be some kind of insects. Yeah, so this is not a fungus, but seem to be some kind of insects that make that, and maybe they lay their eggs into those fluffy things here. Yeah, so this looks a little bit like I don't know, like silk almost. Yeah, very sticky. Because look at this here, and afterwards the microscope was completely, I don't know, yeah, I had to clean it off. It was very sticky stuff. Yeah, banana leaf mite. Could be. Yeah. Did you compare this USB microscope with a mobile phone? Well, what I've done is I connected this USB microscope with a mobile phone. Okay, so I did not. There are some lenses that you can clip on a mobile phone to get some kind of macro, but this one actually works differently. It's actually directly connected to the mobile phone, like I've done over here. Yeah, so you see that. I'm holding the USB microscope, and then it's directly connected. To an Android phone. Yeah. So, you know what I'm gonna do? Let's have a quick look at the stereo. Okay, doesn't look like white flies. They look like tiny white moths. Okay, yeah. So this is again, this is now the actual live image under the microscope. Okay, and I'm just going to zoom in a little bit more. One of the things that I did notice here, and that's also a nice, interesting comparison between a real stereo microscope like I have and this tiny USB microscope. If you just look at this, you're probably going to realize, well, actually, the depth of field is not quite as high as the one of this one over here. This one actually subjectively seems to give actually a slightly sharper image. That's kind of weird, you might think. How is this possible? And the reason is the following, because the diameter of the lens in front, which you cannot see now, is significantly smaller. And the smaller the aperture, the higher the depth of field. So this is the reason why it looks a little bit fuzzy under the stereo microscope. Yeah. So this is kind of just something that you have to be aware of. So you have to do some image stacking. It's not necessarily always a big disadvantage, but this explains a little bit why. With a stereo microscope, it looks a little bit fuzzier because the depth of field is lower. Yeah. Somebody already researched ever smaller. I think the white furry critters are these. Okay. Interesting. Yeah. So this is So let's have a look as well. I've seen a few of these. I have seen a few of these spines here already. And you can actually go through the whole thing here and dig through it a little bit. Yeah. Again, here you see a little bit the limits of the camera. Yeah, see, it's completely white and overexposed, so you don't see any details. And the reason is because when there is a big contrast in the specimen, like for example a dark sand in the background and some light sand grains, what the camera will automatically adjust the exposure to make the sand grains, the dark sand grains, appear sufficiently bright, and then this causes an overexposure of the bright areas here, right? So that's again an issue of limited depth of field. Yeah. So sand is always very interesting. So I just want to show you before every now and then I check also the time a little bit, and some of you probably already watched my other video that I published a few days ago, because I did buy myself also. Look at this here. This here, some sand from the tourist shop, right? Yeah, if you're interested, you can watch the other video as well. I'm just gonna show, for those of you who didn't, I'm just gonna put this here as well, because this is a nice example, because this is not real beach sand, right? Because as I told you, it's not allowed to collect sand. So this is actually imported sand, even if there's a sticker on the bottom that says it's imported. It cost me €7. Still a nice souvenir. Tiny nice little seashells in here, which I have never seen on the beach there. But when you put this sand under the microscope, look, I'm just gonna put this. You're going to see, okay, here we go. I need to zoom out a little bit. Okay, need to focus a little bit. Look how very different it looks. Yeah, very dark, very uniform, and the edges are very rough. So this is a not natural beach sand. This is crushed rock. So it's artificial sand. Right, so they're kind of selling artificial sand as a souvenir. I still do think that it must be of volcanic origin, not only because of the color, but it does look kind of shiny and glass-like. Yeah, yeah. No, not $70. €7, $6.90. Okay, not $70. Maybe I misspoke. And I kind of already anticipated that. Just by looking at it, I already could see that this is not quite real. Yeah, yeah, the thing that kind of surprised me a little bit is I just show you here under the desk here. Let me, yeah, here it is. Yeah, in Spanish it actually said over here. There's a sticker in here, and it says actually it's imported. Yeah, so. That's, yeah, €7 for a little bit of a nice little, it's still a souvenir. Who cares? But again, I did some research, and indeed, one of the reasons is that there are certain laws and regulations concerning the exporting of sand. So I'm just saying this is a little bit somewhat of a curiosity. Yeah, but you know what? Why not? Why not have the following, just for the fun of it? Because we already have it. Let's take out one of these guys here. Let's check if at least... I was already, honestly, I was saying, maybe they're 3D printed. But I think they're not. Yeah. Yeah. So... here we go. They still look pretty regular and nice, but that's... But I don't think that they're artificial. I mean, this one, because some of them do have some tiny imperfections, as is normally the case, or should be the case with natural specimens. Let me try this one over here. Yeah, this still yeah. Yeah, so there is a comment here. I meant comparison of the USB camera with a camera built into a mobile phone. Computers and mobile phones enhance specifically the quality of the image. So basically, if you want to kind of maybe take an image using directly the mobile phone camera, the thing is the following. You don't get the same magnification. I mean, that's, I think, the main issue here, right? Because those tiny cameras, they can actually magnify quite a bit. Okay. Yeah, so this is, I would say, that it would be one heck of a 3D printer to be able to have that type of resolution. Yeah, sure. Without support. Yeah. No, these indeed appear to be authentic. So, but before I move on to the next topic, I do want to show you something else, something that I kind of discovered here. And I'm going to now use this here. Because I did discover that when you turn it, it says here 40 times to 1,000 times. It's of course unrealistic, right? But there is the interesting thing. I thought that this is only a focus, right? But the interesting thing is that indeed, if you turn it, then there are also two different magnifications that you have. Just want to show this to you. Now, not using the... I mean, why not just try it with the sand as well? I might have some sand here on the front. But let's try this, if this works. Let me turn on the... Yeah, okay. So, yeah. So I'm now, I'm going to go now, this is now basically, yeah, I'm turning now the wheel. So I'm touching now the sand, right? And now I'm turning, and you see that this goes now into focus, and it looks kind of okay. Honestly, subjectively, it looks, I don't know, probably even better than, yeah, directly through the stereo microscope, right? Yeah, and then I continue turning it, it goes out of focus, and it goes into focus again. This is something that I kind of consider kind of interesting. Yeah. So, and if you, and of course, now I'm lifting it up, and then you can, of course, also try to refocus it, yeah, when you lift it up, and then you can get also, of course, a smaller image this way, right? So there is, yeah, there are plenty of things to kind of play around with here. Yeah, 2 focal points. Yes, that's indeed the thing that kind of surprised me a little bit. Yeah. I also accidentally discovered this, and I only get those 2 focal. So I don't know how they're doing this. They are indeed, yeah. I can try it here also with my hand. Yeah, I'm putting it against my hand. Can actually see, you know what? Maybe the hair on my hand should be easier to see. I continue turning it, and then I'm getting a second focal point. I in the world don't know how they're doing this. Yeah. So this is kind of, I don't know. There must be some, yeah. It's kind of interesting. Yeah. What you have over here is, let me turn off the light here. You can turn on, yeah. You can actually see this part here. When you turn it, you can actually see that it's going in and out. And I kind of thought, yeah, that's just the focusing thing, right? But it actually also changes the magnification. Huh. Yeah, so this is kind of fascinating. Just I discovered this by accident. Okay, so let's have a quick. I'm quickly looking here if there are any other comments. Industrial resin printers are surprising, and to paint them after you can easily hide any sign it was printed. Wow, we're really that would be, honestly, I don't know. Maybe, who knows these days? I don't know whether these are some kind of resin printers, whether they actually made that. I don't know. I mean, that would be really fascinating. Last try before I move on to the next topic. I don't know. I thought that I've seen some imperfections here. I don't know. But I mean, they do look kind of pretty. I don't know. They do look pretty good. Just for your info, I've seen some souvenirs. Some volcanic rock that they've sold in the souvenir shop. I don't know, 2 tiny turtles made of volcanic rock. And you know, volcanic rock is very porous. It has those holes in it. But if you just compared the holes, they were exactly on the same place. So it was clearly molded from something, or also kind of maybe 3D printed. Yeah. But that is an interesting thing. So maybe I'm going to have a closer look at those here, whether indeed they're authentic or not. Okay, yeah. So I'll kind of move on. Okay, let me quickly read if there's some more comments. Yeah. Liquid UV resin paint printers have amazing resolution. Yeah. Wow. Yeah, I mean, one way of testing it is, and I don't know if I want to do that, is of course I could try to put some acid on it and see if it starts foaming, because seashells, authentic seashells, are made of calcium carbonate, and therefore they dissolve in acid. And the foaming, that is, would be the how do you say, carbon dioxide being produced. Okay, so let me just quickly check the time a little bit. Yeah, so let's move on. Okay. I don't know, changing topic maybe. Okay, because yeah, I just want to try this here as well. This here, because I do also want to show you a few other things, but I did make a couple of more videos also on Tenerife, so I'm going to upload some other videos as well. So what I have here, this is a little bit of how do you call this, a yeah. An add-on to one of my previous videos that I published, and that is the one about the kitchen sponge. So I made a video some weeks ago where the kitchens I looked at some bacteria under the kitchen sponge, and these bacteria were, wow, there were so many of those. And I said, okay, I want to make now a microscope slide, and I would like to stain them. And what I've done, therefore, I've taken some a drop of this liquid from a kitchen sponge and put it on a microscope slide, like here, and I simply allowed it to air dry. And you see that there is, of course, a spot here. I've got another one over here as well. Okay, you see I've prepared another one, just yeah. And what you have to do then, if you want to stain bacteria, you have to make sure that they're kind of baked to the glass slide. You call this heat fixing. Okay, and this is what you can do. I mean, I've got also an alcohol burner, but you can also just use a lighter and then you basically heat it up. How hot should it be? You should still be able to hold it in your hand without actually burning yourself. So it should be fairly hot, but you don't want to burn the specimen. And of course, you do that from the bottom side so that the flame does not touch the specimen. So it's important. This has to be completely dry first, right? And then you do that. And then what happens is when you heat fix it like this, okay, then essentially the cells become baked to the glass surface, okay? You have to wait until it cools again. It's a little bit like an egg sticking in a frying pan because the proteins somehow they form cross linkages to the glass. Yeah, and this kind of sticks now, so that they're not washed away by the stain. And what I want to do now is I first want to show you how it looks like when it's stained and when it's not stained, right? And then I'm actually going to show you the staining process itself as well, obviously. But I just want to show this to you because there is quite a remarkable difference. I need to get again to the scope view. It takes me, yeah, so that's let's just look at this is how it looks like non-stained. I'm going to go bright field, okay, because that's actually what most people have. It's a pretty big mess. We go up with the magnification. Okay, and you see it's actually nothing really, I don't know, yeah, a little bit like expected. And the reason is also because those cells, they do have some kind of a high refractive index. It's not it's basically dry. It's surrounded by air, right? So yeah, all of those round dots that you see here, some kind of maybe some remains of some food particles or whatever was in the kitchen sponge. But actually the bacteria that you see are actually those tiny things here in the background, right? So you see that there is something there, but actually it's not really nicely visible. Okay, so I'm going to show you now how the whole thing looks like after staining. So I'm going to move over. And this is how it looks like. I have to refocus again. It's a little bit too dark, I would say. So let's go to an area which is a little bit brighter. Let's turn up the, yeah. And then what you are able to see now, you're able to see the individual bacteria, those tiny little dots, much better. I'd have to zoom in a little bit more, and under the microscope I actually see it better, but you can actually distinguish the different bacterial shapes much better, right? So for example, over here you see there's some long ones, the rod-shaped bacteria, yeah, and so on. Yeah. So this is kind of yeah, and I've stained this now with you can try methylene blue. I've tried now crystal violet because it's a very extremely strong stain. Yeah. The problem with this specimen, however, is that the concentration of the bacteria is too high. Yeah. You see how many of them are clumped together, right? So in the future, it kind of shows a little bit how many bacteria there are indeed in the kitchen sponge. Yeah. So I should have probably diluted this a little bit. But staining itself, I'm just going to show you very quickly the staining principle. Okay, so I'm going to go back here. So this is a little bit of a follow-up video of this kitchen sponge video, and I'm just gonna use crystal violet because just happen to have it. And what you do is you put a small drop directly on here, and you kind of spread it around. And I don't even know, do I have? No, I forgot where I put the and usually a minute is enough. Yeah, see, it's not everything is properly covered. It doesn't matter. Yeah, there's still something a little bit on the edge. But you know what? That's enough. It's actually a minute or even less than a minute actually is enough. Yeah, and then you do it like this, and you simply rinse it here with some water. And see, even if you put the water directly on the bacteria, they're baked to the glass slide so that they don't even wash off anymore. You see, it's already stained. Then essentially what you do is you dry it, obviously. Normally you air dry it. I'm a little bit impatient, so what I'm doing is I'm just removing here. You can actually also, if you don't rub it off, then you can actually just press it against here as well. Okay, and I'm gonna put this also under the microscope again. Scope view. There's no water on here now. Yeah, and here we go. Okay, you see, the density is so extremely high, the bacterial density, but all of those tiny dots that you see here in the background, these are the individual bacteria. Yeah. So this is the thing. Yeah. Question of off topic. Any idea how to make an at-home diluent for human blood and or for human semen to dilute the sample without throwing osmotic balance? 0.7%. Weight percent, 0.7 of salt water. I think 0.65 or 0.7% is basically physiological saline. So this means 0.7% means 0.7 grams of salt plus 100 milliliters of water. Okay, if you do that, then you have basically a salt concentration that should be okay to mix with blood without throwing off the osmotic balance. If I remember. The reason why I remember this is because this is the concentration that we used at the university, also for other cells. If I 0.65 or 0.7, I forgot. Probably would not make a huge difference. Just for the fun of it. Where is it? This is something I'm not gonna do something which something I'm now going to do something what I normally don't like doing. Okay, I'm going to actually do now oil immersion. Okay, and it works like this: oil immersion microscopy with heat fixed stained slides. You do not use a cover glass. Okay, so that's the thing. Is it possible to make a higher density of bacteria with a centrifugation? Yes, of course. That's what I've done as well. But then you won't be able to see anything under the microscope because the density is so high. So, of course, you can centrifuge it. And this is indeed something that is done to separate the bacteria from the growth medium. So I'll be adding a little bit of oil now. Okay. You might want to watch a little bit what's happening over here on the top. This here is now the how do you say the oil immersion, the objective. This is how it looks like without immersion oil. I'm going to now do the following. I'm going to focus lock. You cannot see that. I'm now lowering the slide. I put a drop of immersion oil now directly on the bacteria. No cover glass. I'm raising it all the way until the focus lock. I'm rotating it back and forth to spread the oil, and I'm focusing. Okay. Now you can actually, at this magnification, that's now basically my 100 times oil immersion objective, we can now actually see the shape of the bacteria quite well. Okay. So very typical, you can look at some of them. Those tiny lines here are rods. Yeah, the round ones that you occasionally see are cocci, and so on. Yeah, so this is actually a just a short demonstration of how you do some staining of bacteria. Yeah. Some time ago, I also did some gram staining where you can actually see that some of them are red, some of them are blue, but just for general staining, this also works. Yeah. So, that's one of the things that I kind of you just look. I mean, look, I still consider this very impressive. How many bacteria there can be in a kitchen sponge? I mean, this is absolutely crazy, right? But again, if you look carefully, you see the individual the rods and so on. Yeah. Of course, if you now look through the microscope itself, directly. You see it even better because here right now we only have it on video. Yeah. So let me quickly check again what's going on here. Okay. Yeah, I'm just reading. Yeah. Okay. Just wanted to share that with you. Let me do the following. Every time after I've used the how do you say, the oil immersion objective, you kind of lower it again. Then you remove it again, the slide, and you carefully wipe off. Now the thing is the following. I'm using synthetic immersion oil. Yeah, so this basically means some folks in the lab, they don't wipe it, they don't clean it because they're using it every day anyway, right? However, I don't want to do that because if there is some oil sticking on there, then of course this can also collect the dust, and I don't like that. Yeah? And if you're using natural immersion oils, they have this yellowish color. You should definitely clean it off because they do have the tendency to turn hard and to solidify over time. Yeah. So let's put this back again, or a lower magnification. Okay, and we're back here at the desk. Yeah. So just wanted to share that with you a little bit on how to look at bacteria. You can try different types of stains like methylene blue, maybe even fountain pen ink works. Didn't try that yet. Yeah, with bacteria, but crystal violet is being a strong stain, or should definitely work. And what I'm doing with the slide next is I have to try to kind of wash it relatively aggressively to wash off the bacteria again. Yeah. Can contaminants be removed by filtering so that only the bacteria remain? Basically, what you... Yes, it depends on the filter. So it depends also what you mean with contaminants. So what you do is you can use different filters, and then you kind of remove any larger particles as well. So yes, they do sell different types of filters for that purpose. However, sometimes what you also do, especially when testing water quality, you're filtering the water, and then you have the bacteria on the filter, and then you analyze those bacteria. So you're actually going the other way around, right? So you kind of you want to make sure that the bacteria are in the filter. Of course, you also have other particles on there, but then you kind of try to analyze those bacteria that are on the filter. Yeah. Staining and phase contrast did not really work. It doesn't look good. Just because you're asking, because I'm just going to So let me quickly show this to you again. I'm going to put this back in, because somebody asks about staining and phase contrast. Not always a good combination. Okay, so I'm just going to show you very briefly. Let me see if this actually... So So, okay, so that's now, okay, I'm going to show you now. So that is now, let's bright field. Okay, so you see it's stained. And now I'm going to switch over to phase contrast just by putting in the phase contrast annulus. I have to open it. And this is how it looks like in phase contrast. It's some kind of an, you see it's some kind of an overlap, right? Because the background is dark. Yeah, and you see you're kind of mixing the phase contrast with the actual staining, so that kind of defeats the purpose a little bit. Yeah, and that's again bright field. Yeah, so you see, you gotta try it, but generally that's not what you want to do. Phase contrast you want to use with transparent objects that are not stained. Yeah, so this is a little bit the thing. Yeah, so let's move on. Okay, how is that? Let me quickly turn this off again and back to the desk view. What else is on my plan today? Bacteria stain. Oh yeah, the thesis and the bird bath, and then maybe some Q&A. That's the thesis. So some theory. I just thought I need to clean this a little bit, otherwise there's I don't want to dump any stain or ink or immersion oil or sand or anything or water sample over my thesis, because it's the only copy I have. Seriously. But I just thought I'm going to share it to you with you in any case, because there's a little bit of bacteriology, and some folks every now and then, in the comments section and so on, they're kind of asking me things that are a little bit related to that. So I said, well, I'm just going to show you a little bit of what I've been doing. So it's actually not thick, the book. It's around 90 pages. So almost 2 years of lab work. It's a master's thesis, right? And the significant part is actually written in German as well. But the back part is also written in English, and I just want to show you a couple of things what I have done here. And the pictures are horrible, because this is a photocopy, and this is over, I don't know, from the year 1998, so it's over 30 years old already. Yeah. And basically what I've done is, here's some photocopies of the bacteria. It's all phase contrast. At that time, digital photography did not exist yet. So basically this was taken with an analog film, and then I scanned the film so that I could include that. Just want to show with you what I've been doing here. Let's have a look at those this data here. And normally these days you probably wouldn't do this anymore. Yeah, because you just would do a DNA test. But these are basically the bacteria that I numbered. That's my designation here. And what I've done is I've had to grow them, and I had to basically then test whether the certain enzymes are present, like catalase enzyme, oxidase, and so on. And basically do a whole range of biochemical tests on those, on whether they're able to metabolize or do certain things. Yeah. For characterizing them. Then over here, I did a whole bunch, again, I did a whole bunch of analytical chemistry. The presence of so-called quinones, amino acids, the main fatty acids that are present, polyamines, and so on. And based on this, the combination of the chemical composition of the bacteria and a lot of research on papers, I could kind of estimate into which bacteria they are. Why am I saying this? Because I'm getting a lot of questions every now and then. It says, I found a picture of a bacterium under the microscope. What is it? And the bottom line is you cannot say that. So if you look at the bacteria that I just showed you under the microscope, you would say, okay, you see some kind of a bacterium. What is it? You cannot say it, right? Microscopy, unfortunately, is not able to provide that information. Yeah. So what you have to do is you have to actually do a biochemical test. The example that I'm always giving is you cannot determine the brand of a car by looking at its color. It's not going to work, right? Because color is not a specific distinguishing characteristic. Yeah, I cannot, find out who am I talking to by what's the name of the person by looking at the hair color. Right? It doesn't work because hair color is not highly specific, for a specific person. And that's a little bit the same thing when you're looking at the shape of bacteria, which I consider a little bit ironic, because microscopes are so important for actually microbiology, but then at the same time so extremely limited, right? Because the actual things that if you want to know which bacterium it is, you got to look deeper, right? And so this basically was a lot of work. And I say now unnecessary work, because nowadays what you're doing is you're doing just a DNA test. Yeah, so that is a little bit of a thing here, and there was not a single database that allowed me to determine, okay, if it's got the combination of these characteristics, biochemical then it's that bacterium. It didn't exist, so I had to really dig through a lot of and it goes on over here, right? Yeah, which certain chemical substances were present. Yeah, and then based on that, I could estimate probably which bacterium it is. So, and then over here, these are basically all of the fatty acids that I extracted. So I did a lot of analytical chemistry. So basically, what I've done is I've grown the bacteria, and then I did some biochemical analytical chemistry tests on those bacteria to identify which substances are present. Yeah, and then based on that, you could try to estimate what it is. Yeah, and here in the back part is also an English part, yeah, where it summarizes. And then something that I just want to show you. These are the references. I wonder where it is. I also did some DNA sequencing, and that's of course the why is there not where's this picture? Maybe it's over there somewhere. Yeah, there is. You can also establish then some so-called those trees here, right? So this was actually the internet. I mean, this was 1998. I mean, the World Wide Web was just invented. I don't know what 5 years before that. So I was very happy that there was some kind of an online database search. It was a very primitive program, but essentially it allowed me to compare basically my results here of those bacteria to other bacteria in the database, and then the program established some kind of a family tree. Yeah, so that's basically it allowed you to put your or my bacteria into relation to other bacteria in the database. Yeah. So this is kind of the thing that I was doing. And these days, what you do is you don't do all of those biochemical tests anymore. You basically do a DNA analysis right away. Yeah, I mean, or for example, this strain over here, just by looking at it, you see it's a rod shape, but you cannot say simply by looking at it what it is. Yeah, so that's a little bit the fascinating thing. And honestly, I don't know. If I were to turn back the time again, I probably would do a few things differently. Yeah. But yeah, you of course with time you learn a little bit. Yeah. So all of those. So for example, this here you see that it's very bright. So these are probably some endospores that were being formed because they usually shine up very brightly in the image. And look at this. These are all photocopies, and the image quality is horrible. But still, yeah. This was a super expensive phase contrast microscope at the university. Yeah, it was hugely expensive. Yeah, but I just wanted to share this a little bit with you. On how basically biochemical analysis can be used to estimate the bacteria. But then I did some research recently, and it's not done anymore, right? Because this is way too much work. And you basically get much better results or much faster by simply doing a DNA analysis right away. Yeah. Has my work been used anywhere, perhaps as a professor in his thesis? I know that there were 3 publications that resulted from that, where I was a co-author. It's what I know, it's in the library of the institute somewhere, and also all the theses are in the National Library. I don't know, honestly, if it's been used. I personally learned a lot in the process, that for sure. But it was a lot of work where I feel in retrospect, from a scientific value, I would have done probably more DNA analysis. Yeah, just a couple, I mean, basically bacteria that are isolated from the air, to do a general air quality assessment. honestly, more bacteria would have been better, and spend less time on the biochemical analysis and more on the DNA sequencing. I think you would have gotten a better overview of what's actually present. On the other hand, I learned much more in this way. Yeah, because this was actually quite a bit of, I learned a lot of different analytical techniques. Yeah, were you impressed with your findings afterwards? No, I wasn't. I'm not impressed. I mean, the thing is the following. It's very easy to find new bacteria. The reason is because most bacteria have not been characterized yet. Right, so so finding some kind of new, unusual combinations is nothing difficult. Yeah, it's almost to be expected. Yeah, 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 yeah. The info is very useful in certain areas of research DNA for phylogenetics, but phylogenetics is not everything. Yeah, I agree. Right, so if you want to characterize bacteria a little bit more of what chemical composition they are, of course, this is called chemotaxonomy. Yeah? But the difficulty was for me is from the combination of characteristics over here, how can you basically yeah, go to the, yeah, what I've done here, this is the DNA, yeah, based on DNA, and sometimes I had to kind of cross-verify whether the DNA sequencing results were the same, more or less, of what I got over the biochemical analysis, yeah. So this was a lot of how do you say, headache. But I think what maybe the bottom line is, and that's kind of what I want to share with you, is that if you want to actually know what bacterium it is, then in many cases, yeah, you have to go. The shape or form is not sufficient. Now, having said that. Sometimes when you're testing the drinking water quality or swimming pool quality and so on, you don't have time to do all of this stuff, and it's also not necessary. So if you want to simply test the quality of certain water, swimming pools or lakes or whatever, what you do is you basically quickly take a water sample and you filter the water and you simply determine the number of bacteria, and then essentially, if the bacterial count is too high, then you can assume that the water is polluted. Yeah, you don't even need to go into the details of finding out what bacteria they are, because if too many bacteria are present, then you know it's already a problem. Yeah, so that's a little bit the idea here. Okay, well, I just wanted to share that with you a little bit. I don't know, yeah, just because I also, after so many years, wanted to have a quick look at what I've done at that time. So let me quickly go through the comments again. Yeah. Yeah. Okay. Do you want to see the unstained bacteria in phase contrast? Okay, that's another thing. Where did I put the slide? Oh, I need to. Oh, here is it. Okay, there are some questions. Thank you. Thank you. I do have another slide with unstained bacteria here. I can put those into phase contrast, but be aware that there is now no water on it. This basically means there is no, it's not surrounded by a medium, so that's so. This is basically how it now looks like, the unstained bacteria in phase contrast. You see a lot of other stuff here, I don't know, some food particles, yeah. But the bacteria can be seen here now, again, as darker structures. Okay, so ignore all this stuff here. These are some other, dried up things here. Probably washed away during the cleaning process, but those, dark things in. But you know what? What I'm gonna do? Because you're asking, I'm going to do the following. Because I said it's now surround. Let's try the following. I'm going to add a little bit of water. So I'm going to make a wet mount, and then let's see how this actually looks like. Yeah, if the contrast improves. Okay. So before that we tried it with oil immersion, right? I cannot use oil immersion on my phase contrast objective. I'm going to remove this again. And where is the desk view again? So I need some water. I need a little, just a second. I need a beaker. The wrong beaker. Gee, I'm super well organized today because all of the other beakers that I have are a little bit dirty. Oh, here, this one is fine. Yeah, that's okay. So let's do the following. Let's see how this the unstained bacteria, how they look like in phase contrast. But now I'm going to actually add a water on top so that the difference in refractive index between the bacteria and the air, so there is no surrounding air. So let's have a look now at this here under phase contrast, if this actually looks better or not. Of course, now you see that many of the bacteria are actually being washed away because I did not heat fix them. See, on those round things seem to be kind of fat droplets. Yeah. So you actually see that heat fixing actually might be a good idea. But let's just ignore those moving droplets here. And you see that those tiny things here in the background, these are the bacteria. Okay. So just because I also wanted to know how this looks like. Huh? How can you calculate how many bacteria there are? If there are too many bacteria from a water pool, do you count the bacteria in the field of view or in the square micrometer? Okay, yeah, so that's actually an important question. If you want to do water quality assessment, you cannot use a microscope. Why? Because I read somewhere, depending on the type of bacterium, 100 bacteria per milliliter is already a critical level, and that is too low to be seen under the microscope. That's the problem. Yeah. So here we have an extremely high density of bacteria, and we can see them, all of those tiny dots that you see here in the background. But for water to be safe, for drinking water to be safe, you're not even allowed to have one coliform bacteria per milliliter. And I think for swimming, I think the critical level is around 100 bacteria per milliliter. A milliliter is a cubic centimeter. Yeah, that is, so it's possible that you take out a sample and you put it under the microscope and you won't even see it because there are too few bacteria in there, but it's already above the critical level. So what you have to do is if you want to test the water quality, you have to actually take a large amount of water, let's say 100 milliliter or a jar of water, and then you have to filter it so that the bacteria are on a filter, and then you start to grow the colonies on the filter so that you can actually count the bacterial colonies. And then you do a count like that. Yeah. So you cannot use, unfortunately, you cannot use a microscope. Yeah. So this is so what you have to do is you have to actually grow the bacteria. Yeah. Okay. Did you manage to get any samples while you were on your vacation? No, I did not take along any samples otherwise. And the USB microscope is not going to allow me to to look at very tiny microorganisms. Yeah? Is 100 times water immersion used for bacteria in phase contrast, or how do you most clearly see them? So what you can do, and this is what I have done, is you can use, and I don't have that on my microscope, but you can indeed use 100 times phase contrast oil immersion objective. Okay, and that's actually what I've used at the university, and that gives you very good images, right? Of unstained bacteria. Yeah, it's 100 times oil immersion. Yeah, but it's also phase contrast. Yeah. Can you use a counting chamber? Same issue. For water quality assessment, no, because the number of bacteria is generally too low, but it's still above a critical level. Yeah. Okay. Yeah. For live bacteria, yes. For live bacteria, and that's what I've also used. I've also used, in this case, for live bacteria, what you can do is the following. You take a live sample, you use very little, you don't even heat fix them. You put a cover glass on top. In this case, you do have to put a cover glass, and then you use oil immersion. Yeah. So that is, yeah, Marie was asking. Yeah. Yeah, and cytometry also works with live cells. Yes. So, having said that, I'm checking the time. I'm running a little bit over time, but I'm going to do the following. I'm going to, before I answer a few questions, let me put this away. I've got slides all over the place now. Where is the desk view? I'm going to quickly show you somewhere. Yeah, the Hematococcus. Let's see if this is the one. This is basically over here. You can, yeah, I've collected the sample and I added a little bit of the stuff that's down here. This is from a bird bath, just because, so that we can actually also see some live samples today. And where is this scope view again? Ooh, I'm still in phase, contrast, and dark field. So let's go up a little bit. And I found some interesting larvae again. Let's put this away. And yeah, so maybe another one. I did find a couple of rotifers again. Yeah, so what you see over here, those round things here, these are called Hematococcus. These are photosynthesizing algae, but they have a red pigment, and they're very common in bird baths. Yeah, and yeah, I just happened to be next to the bird bath, so simply took along a sample again. There was actually more movement going on before. Maybe it's already a little bit dried up. Here in the center right now you have what looks like a vorticella. Yeah, and all of those round things are Hematococcus, and I don't know. This is not a very interesting. I think the problem is that this sample seems to be already pretty dried up. So what I'm gonna do now is let's try it again with a new Somebody also asked me recently about my opinion concerning so-called concave slides. They have an indentation, and whether I use those, and it says occasionally I do use them, but I do not consider them absolutely necessary. So what I usually do is I want to have a quick look, and if I do not want to inhibit the movement, then I simply just put a drop here and I look at it under low magnification, simply for first inspection. And scope view here. Okay, I need to refocus again. Okay, yeah. And you see over here a lot of what seem to be some kind of, I don't know, of water fleas or some kind of crustaceans, the exoskeleton. Yeah. If you want, I tried a concave on the first fair shrimp nauplii and it crushed it immediately, yes. Sometimes what also happens is what you might want to try is either some kind of spacer rings or to use other cover glasses for keeping a distance. Yeah. Yeah. So that's basically the typical appearance of a bird bath. I used to find just before when I looked at it also some rotifers again. This is which I consider interesting right now because there seem to be fewer around right now. I think it depends also a little bit on how long the water has been standing, if it's been raining recently or not. Yeah. But in any case, I can recommend if you find not only bird baths, but generally if there's some kind of water collected at a place where there's some kind of a rock, right? Then you very often you find those those round algae. Okay, but let's go over here again onto the other side because I do want to see, because before there was actually, let me put, there was actually some kind of an insect larva somewhere. Yeah. So usually a lot of exploration is possible. Here's some dead insect parts as well. Yeah, here's something moving around. Ah, here it is. Okay. Maybe already a little bit crushed. Yeah. But those are very typical of insect larvae. Ah, it's still alive. It's a little bit crushed because maybe let's add a little bit more water. Maybe this will give it a little bit more. Yes. So it's now got a little bit more freedom of movement. So this basically, yeah, and on the back side, you see the breathing organs. So possibly some kind of, I don't know, maybe a mosquito or related. Yeah. So this is midge larva, exactly. Yeah, very common. So that's actually one of the reasons why you want to try to avoid standing water, because if it happens to be a biting midge or a mosquito, then of course you've got the whole issues with diseases and all these things, you know. Yeah. But so now basically you see that always interesting things. So normally what I would do then is when you kind of let it stand, then sometimes you can see after a couple of days, you can actually see that there are then tiny little insects flying around in here. Yeah. Yeah. So what I'm going to do now, I think I'm just going to move on a little bit. Yeah. So you see that there. None. Here it is again. So actually, if you're limiting the water, then of course this makes observation sometimes easier because the objects and the specimens cannot move around quite as quickly. Wow, what's this huge thing here? But then, again, sometimes, this here is rotifer. Not quite as many as there used to be. So you have also here a natural progression of organisms. So let's do the following. I'm going to now, for the last couple of minutes, go quickly through some of your comments, of which I probably have already answered many. But maybe there's some more. Yeah. I'm just. Have you seen the Hematococcus in the motile stage? Yes, as a matter of fact, several months ago, I've actually seen some of them being motile, and moving. Would you say that the beach is safe? I don't know if this is a question for me. Let's put it this way: if this refers a little bit to the mosquitoes and to the midges and so on, these are fresh water, right? And normally you don't see them actually in an ocean, right? But if you've got somewhere standing water, flower pot water and so on, then you should actually be, and a little bit, watch whether you're kind of growing mosquitoes or not, yeah? Oh, I mean the amount of bacteria and the kinds. Okay. Separate story, because you're asking. When I was and I'm making a video on this. When I visited the Canary Islands last week, actually one of the beaches was closed. They closed it because they did microbiological tests, and we went there, and right as we went there, they closed the beach. Right, they put up signs, swimming prohibited. We asked, and it turned out that essentially they found out that the bacterial concentration of sewage was too high. So it was not safe for swimming, which kind of surprised us a little bit. And then later on in the news, I read that the very next day it was open again. It was considered safe again. And what they said actually is that somewhere else, off at a different place, because of the currents and the tides, contaminated water was flushed onto the beach. So my opinion about this is the following. The good thing is that in many tourist areas, they are doing microbiological testing to actually make sure that the water is safe. Yeah. So especially when you've got many people going there, it's an important public safety measure. So and that's what I trust. So they're doing regular testing of the water, and if the water quality is not good, then they're going to close the beach. Yeah, so that's in that sense, I'm not so much concerned, because essentially, if the authorities are taking care of the whole thing, and if they've got a microbiological laboratory, then this is part of the regular public safety and health measures, right? Yeah. Yeah, nothing ruins a beach trip more than sewage. I totally agree with you, but there it was like this, that the water looked completely clean. What shall I say? I mean, but still the bacterial load was considered too high, and indeed this was also reported that I checked in online that indeed they had to close a beach. And the funny thing is, not funny, but I mean interesting thing is that the very next day it was open again, right? And what they said is indeed they assumed that because of certain currents and tides, that some sewage that was somewhere in a different place was kind of flushed onto the beach. So yeah, very quickly, I'm not going to go into all of the details here, because some of them I already mentioned, okay? Mass cultivation of Haematococcus. You could always line up topics in a queue. If there isn't enough time, then continue. That's actually also an idea. I have to say the following. One of the things that always stresses me out a little bit is coming up with ideas for the live stream. This is always a slight stress for me, because sometimes I know I'm repeating things that I've already done before, and are you interested? Are you not interested? I try to find variations. So that actually might be an idea, so I don't pressure myself so much. Yeah. Did you know it's illegal? Yes, I knew that, and some tourists actually got in real trouble because of that. Okay. Yes, please do visit some YouTube channels. Okay. I had some microscopic samples I found in amber. Yep. I would say the challenge here is because amber itself also is not flat, but also sometimes curved, whether it's possible to properly focus through the amber. That would be because the amber itself also acts a little bit like a lens. So one would have to try that. Never tried it. Yeah, the white thing, these were some insect cocoons apparently. Okay. Thank you for showing us the black sand. I suppose the colored bits are mostly glass, or that what else could show up? Yeah, sometimes, I don't know. Of course, there could be some kind of transparent rock and quartz material, but I indeed have seen on some beaches quite a bit of glass. Which are actually with fairly sharp corners, which actually indicates that this was kind of recently scattered. Yeah. 1,100 microscope. Don't know that. A microscope. I'd have to look at it. Yeah. Does the USB microscope have a threaded connection like a camera? Then you could use a small monopod as a hand rest. I've had. Yeah, it actually did come along. The USB camera did actually come along with this here. Yeah. Which I consider a little bit, I don't know, not very stable. Yeah, it's not the most stable thing. Yeah, you have to tighten it here, but even then, it's not entirely stable. Yeah, yeah, not certain, but closest match I could find. Okay, yeah, that refers. I recently used a microscope to analyze the white deposit of a local river gravel. Found it to to be cremation ashes. Really? Okay. Calcium carbonate also localized increased pH. How did you know? How did you actually find out that which ashes they are? That would be interesting. Yeah, it wouldn't surprise me, but yeah. But that would be interesting. I meant to comparison of this USB camera with a camera. Yeah. I know I've bought some time ago actually some lenses that you can clip onto the camera of a USB of a mobile phone. But what I find difficult, of course, you need some kind of a system in place to actually put the specimen at a precise location away from the camera lens. And I think that's a little bit of a challenge, right? With those USB microscopes, what you have is because you're kind of holding it like this, right? Therefore, you're automatically always keeping the distance constant, and then you can focus more easily, and this also prevents movement. Right, so you can actually get when you're pressing it against it, you can get actually fairly stable picture. Yeah, with a mobile phone, holding a mobile phone like this it might be a little bit shaky. Yeah, so unless you're someone maybe able to place a microscope slide on the mobile phone, I don't know. Yeah. Ashes, here we go. Ashes predominantly crushed bone, calcium carbonate, so reacts with acid. The container tube. No, of course, then this of course gives away a lot. Okay, yeah. Then if you find the container even, then you know. Okay. Years ago, I looked at a sample of black stagnant water that had been dripping in a sink for years. The things I saw made me quit microscopy for a long time. You know what? If you really want to look at something interesting, look at in your sink. Look at the biofilm. If you take out, if some sinks have this stopper, right, and it's you take it out on the bottom side, it's kind of very slimy. There's this biofilm. Have a look at that. That looks pretty interesting as well. Yeah, the slime. That's a biofilm. It's full of life. Yeah. Off topic. Any idea how to make it at home diluted human blood? Yeah, that's this physiological saline of the 0.67 or 6.5 or 0.7%. Yeah. Yeah, you can use centrifugation. That's actually what I've used myself to actually concentrate bacteria. And contaminants can be removed also by filtering, not chemical contaminants, obviously, because dissolved chemical substances cannot be filtered away. Okay, so that's the thing. Yes, there is a question: is your microscope forum still active? How can one join? I will tell you what the following situation. Yes, it is active. Yes, you can join, but you have to send me an email. I have to manually allow you into the forum because in the past we've had so many spam registrations. So this was indeed a problem. What does this mean? There is a question you have to, I'm asking some simple questions about a microscope. What is the eyepiece called and stuff, so simply that you know that it's, you're a real human being. And there is also this justification. Why do you want to join the forum, right? And then you have to type in some kind of a comment, and then I know that you're a human being. But now with AI, we have the probably... what the problem is with AI, that even AI is able to fill that out, right? The inbox is now, yes, I've now, there has been a problem that people could not send me emails because of one of my, I've got actually two email addresses, editor@microbehunter.com and oliver@microbehunter.com, and one of them was actually full, and I've basically removed now, I've cleaned, so there should be possible again. Yeah, but that's a little bit the thing. So you gotta send me, you gotta sign up, and you gotta send me a quick email with a request that I manually, how do you say, Or another thing that I've been thinking of, maybe if you want to join the forum, maybe I'm going to I'm gonna make people who want to join the forum watch a short video, and I'm gonna give you some kind of a passcode to type in. Yeah, that could also be a possibility. Then I don't have to do that manually. Yeah. Yeah, it was completely full. Yes. Yeah. So that was a little bit the comment here. Let me quickly have a look at some of the comments here. I'm just reading some of the comments here. Lots more microbiology than microscopy. Your other channel could expand into these techniques. Yes, thank you for reminding me. A couple of weeks ago, and I've actually, and I mentioned this also previously already, I've got 2 YouTube channels. Actually, I got a few more. Most of them, some of them are dormant. But one of them is called Microbehunter, and the other one that you are on right now is called Microbehunter Microscopy. The Microbehunter, the main channel, I used to do all of the live stream and all of the microscopy work on there. And I have to tell you that the number of views have crashed down totally, right? For whatever reason. And I also know why a little bit is because I had some viral shorts on there. I had the wrong subscribers on there, and I tried to consolidate everything a little bit. So what I try to do now is I'm going to now make it slightly more, I almost wanted to say dry. I'm going to make slightly. I move all of the microscopy stuff, hands-on microscopy stuff, and microscopy vlogs and specimen preparation and hardware and reviews. I will keep on this channel now, and the other channel, the main channel where I used to do the live streams and the microscopy, I want to do now more some kind of microbiology explainer videos, which are kind of more talking head, a little bit more dry maybe. And this is also indeed where I might talk a little bit more about the more theoretical stuff. So I'm going to try that way because in the past I kind of dumped everything into the main channel and neglected the other one a little bit. So I don't know. Yeah. So I try to reorganize this a little bit, and we're going to see how this is going to work out in the future. Okay. Yeah, I've been thinking about this as well. Some admin rights. Yeah, yeah. Sooner or later, I have to think anyway how this whole thing goes on, because currently everything is on me right now. Yeah, microscopic mystery channel. This is another one. This is several years ago. I have been doing a lot of experimentation. There's one channel called Microscopic Mysteries, which is where I've been thinking about some kind of a stereo microscopy channel because I wanted to promote stereo microscopy a little bit. But it's not been active either. Yeah. Yeah. So, and not only that, there is another one that I experimented called All Things Microbes, where I've been also posting a couple of videos. So you see, yeah, exactly. Yeah, so you see, I've been experimenting around quite a bit over the last couple of years. But right now, I want to focus primarily on those 2 channels right now. Yeah, and we're going to see where this is going to take us. And what I'm going to do now, yeah, too many channels and too much experimentation, and maybe that's also one of the problems. I should have probably stuck to one or 2 channels only, and not always open up a new one. Yeah, yeah, there are a couple more. Indeed, yeah. Not all of them microscopy related, but many of them are actually dormant. I even have one a channel in Esperanto, in the constructed language Esperanto, which I have also not been keeping active for some time now. Yeah, and some channels I've just also got for school, where I'm posting videos for my students, internal videos. But you know what I'm gonna do now? I think I'm gonna call it a quits for today. Okay. Yeah, I would like to wish you a nice week. I'll keep you updated on other things that are going on in the microscopy world that I'm doing, and to make videos, and I'll basically post them in this channel. But also do check out my other channel because I'll be uploading some explainer videos there as well. Enough for today. I already feel a little bit my mouth drying up because of the talking. And yeah, I hope to see you around next week. Yeah, and yeah, all the best, and happy microbe hunting as always. Bye bye.